The first rays of dawn crept through the tall windows of the University of Pennsylvania's aging research wing, casting long shafts of pale light across the fluorescent-lit aisles. In a tucked-away corner of Lab 3C, Dr. Marcus Greenham moved with quiet determination, adjusting the focus on a high-resolution microscope. His dark skin glowed in the cold white glow of the instrument panel as he leaned in, searching for the first signs of success in the viral vector he had spent months engineering. To him, each shimmering particle on the slide represented hope. Hope for a breakthrough therapy, and hope to prove that genius knows no color. 2 years ago, Marcus had arrived at the lab fresh from his postdoctoral fellowship with nothing but a handful of published papers and a head full of audacious ideas. He'd grown up in West Philadelphia, raised by a single mother who worked double shifts as a nurse. Every evening, she'd come home exhausted, but insist he finish his homework before she tucked him in. "Your mind is your quiet rebellion," she'd say. "Keep pushing it." Whether dissecting biology textbooks under the dim light of a desk lamp or coding simulations on a patched-together laptop, Marcus learned early that excellence demanded both sacrifice and courage. Yet, despite his sterling credentials, a summa cum laude degree from Howard University, a coveted NIH fellowship, Marcus found himself relegated to the lab's periphery. The old guard professors, all white and set in their ways, quietly handed him the grunt work, pipetting reagents at midnight, cleaning up data that no one else wanted to touch. He was the first to arrive in the morning, the last to leave at night, and in between, he watched men with lesser qualifications cruise past him for grant meetings and keynote slots. In the coffee room, hushed conversations would, "Did you see his name on that protocol? He's different." Or worse, silent stares, when he cleared his throat to ask a question. That morning, as Marcus jotted observations into his battered leather notebook, the door swung open. Dr. Raymond Bartlett, a senior white lab director, with a reputation for exacting standards and old prejudices, strode in, his loafers clicking against the tile. Marcus snapped to attention. Bartlett peered at the monitor, glanced at Marcus's notes without a word, then shook his head. "You're fiddling with the wrong promoter sequence again, Greenham," he said, voice low but sharp enough to echo off the glassware. "This design is too unstable for an investor pitch. We need something more conventional." Marcus felt the familiar flash of indignation, but held his tongue. He had built this viral vector around a novel capsid protein that, in theory, would target diseased cells with unprecedented precision. His preliminary data showed dramatic uptake in cultured samples, data that he knew could revolutionize gene therapy if given the chance. Yet Bartlett dismissed it as risky, unproven. Marcus closed his notebook and replied evenly, "I understand the concern, Doctor Bartlett. But if we modify the regulatory element to reduce off-target expression, as I suggested last week, the stability should improve substantially." Bartlett's lips thinned. "I'll think about it." Then, without another glance, he pivoted and left, leaving Marcus alone with his hopes and doubts. Once the door clicked shut, Marcus exhaled, rubbing the bridge of his nose. The room was silent again except for the hum of the laminar flow hood. He poured himself a cup of strong black coffee, the bitter taste grounding him. Despite the constant stonewalling, Marcus refused to abandon his vision. Every setback only sharpened his resolve. By mid-morning, the small lab was a bustle of motion. Undergraduates lugging boxes of pipette tips, a postdoc measuring out cell culture media, and a graduate student leveling the bench for afternoon experiments. Marcus moved through them all with calm focus, orchestrating tasks, mentoring younger students, and quietly refining his protocol. Though his colleagues rarely acknowledged him beyond curt nods, the undergrads respected his brilliance, and they showed it by hanging on every word when he taught them new techniques. As sunlight warmed the lab's concrete floor, Marcus paused at the threshold of the exit. He gazed down the hallway lined with locked doors and whispered theories scrawled on whiteboards. Outside, Wall Street investors and venture capitalists would be preparing presentations on sleek laptops, oblivious to the seeds of tomorrow's cures germinating in places like this. In that moment, Marcus felt both the weight of prejudice and the thrill of possibility. He closed his eyes, drew in a steadying breath, and whispered to himself, They'll see soon enough. With his resolve steeled, Dr. Marcus Greenham returned to his bench, ready to turn the shadows into light. The morning sun had barely risen over the Philadelphia skyline when Dr. Marcus Greenham logged into his first virtual pitch session with the prestigious Harrington and Cole Capital Partners. He'd spent the previous week polishing his slide deck, 20 meticulously crafted slides, illustrating the safety profile, targeting precision, and scalability of his viral vector system. His heart pounded as he adjusted the webcam, smoothing the lapels of his lab coat over a crisp white shirt. This was his moment to secure the funding that could transform his concept from a desk-bound experiment into a bench-to-bedside reality. On the other side of the screen, a group of sharply dressed figures sat in a mahogany-paneled conference room, their sleek devices glowing in the dim light. At the head of the table sat Rachel Whitaker, a senior partner known for her razor-sharp instincts and unwavering skepticism. To her left, Samuel Jacobs, a hedge fund manager with a penchant for disruptive technologies, flicked through his laptop, his brow furrowed. Andrew Thorne, the veteran biotech analyst, leaned back in his leather chair, arms folded, ivory cufflinks glinting as he tapped a polished pen against his notepad. Marcus cleared his throat and began. He guided them through the science, the novel capsid modifications that directed the vector to specific cell receptors, the CRISPR-based gene edit that corrected the faulty protein expression, and the automated microfluidic system his white engineer collaborators had helped design for scalable vector production. Each point was backed by preliminary in vitro data, graphs showing over 85% transduction efficiency and negligible off-target effects. When he finished, the room fell silent. A soft cough broke the tension as Rachel folded her hands. "Doctor?" "Greenham," she began, voice cool. "Your data are impressive in isolation. But frankly, I'm concerned about the risk profile. Viral vectors have a history of immunogenic responses." She clicked to a slide of a liver biopsy under fluorescent stain. "What makes you so sure your safety switches will hold up in humans?" Marcus met her gaze calmly. "Our dual fail-safe mechanism uses an inducible promoter that can be deactivated on demand," he replied, pointing to the schematic on slide 14. "In rodent models, we observed complete cessation of transgene expression within 12 hours of administration of the small molecule antagonist." Samuel Jacobs leaned forward. 12 hours? That's tight. What if there's a lag in clearance or patient compliance issues? The last thing we want is a regulatory fiasco. His tone was thinly veiled impatience. Marcus swallowed. He'd anticipated these questions, but hadn't expected the underlying tone. "We've built in redundancy with a secondary microRNA-based kill switch. Even if the promoter fails to shut down, cellular expression of the miRNA will degrade the vector genome. We're currently optimizing the dosing protocol to minimize patient error." Andrew Thorne raised an eyebrow. "Optimizing. That's industry speak for we don't have enough data. Investors in this room expect human safety data, or at least large animal datasets before serious commitments." A soft murmur of agreement rippled around the table. Marcus felt a tight knot in his chest as he recognized disbelief behind their polite veneer. He took a deep breath and pressed on. "I understand your hesitation. That's exactly why I'm requesting a seed investment of $2 million to fund a GLP-compliant study in non-human primates. With this data, we can file an IND and enter phase 1 trials within 18 months. For a moment, the only sound was the faint hum of the conference room's air conditioning. Then Rachel Whitaker tilted her head. "Doctor Greenham, let's be frank. You're asking us to gamble on untested science from a researcher we've never evaluated. I've seen dozens of these pitches, and most flame out. What makes you think you're not going to be next?" Her words stung. Marcus's pulse thundered as he felt the weight of every unspoken stereotype hovering in the pixelated space between them. Yet, he refused to wilt. "Because my team and I have already overcome the initial hurdles. Our White Lab engineer partners, Leah, Ethan, and Hunter, have perfected the manufacturing process. Together, we've reduced batch variability by 70% compared to industry standard. We have the technical know-how and the data to prove it." Silence fell again. Then Andrew Thorne closed his laptop with a decisive click. Thank you for your presentation, Dr. Greenham. We'll review your materials and circle back. The meeting adjourned without a single nod of encouragement. The screen went blank. Marcus sat frozen for a heartbeat, staring at the empty Zoom window. A single notification blinked. Harrington and Cole declined to move forward. His fingers hovered over the keyboard before he closed the laptop and leaned back, exhaling slowly. The bitterness of rejection wrapped around him like ice. Here, in the financial heart of America, expertise and tenacity were apparently no match for pedigree and privilege. Yet, as he rose and stretched his shoulders, Marcus felt resolve solidify within him. If Wall Street wouldn't recognize his genius, he would find another path. With or without their capital, he would prove that innovation and justice could not be gated by prejudice. He shut the lab door behind him, determination blazing in his eyes, ready to chart his own course. Late one evening, as the fluorescent lights in Lab 3C hummed their steady rhythm, Marcus Greenham leaned back in his swivel chair and rubbed his temples. He'd been poring over the latest transduction efficiency curves for hours, determined to squeeze out another percentage point of precision. His laptop pinged with an incoming email, an automated acknowledgement from a Biotech forum he'd posted on days before. Scrolling through a few perfunctory notifications, he nearly dismissed it, until he recognized three unfamiliar names in the subject line: Leah Montgomery, Ethan Price, and Hunter Caldwell. He opened the message with mild curiosity. Each of them introduced themselves as former engineers at a top-tier pharmaceuticals firm, white, freshly disillusioned, and hungry to work on something meaningful. They'd read his preprint on the novel capsid design and believed, in bold contrast to the investors he'd faced, that his idea was not only sound but crucial. Boldly, they invited him to meet at a co-working cafe they'd rented for a small startup weekend, promising detailed sketches of a scalable microfluidic bioreactor array they'd prototyped. Marcus stared at the screen. His fingers hovered over the keyboard. Could three strangers, white outsiders to his world, possibly understand the nuance of his work, let alone the weight of his daily struggle? He closed his eyes and felt the familiar tug of skepticism. He typed a cautious reply. Thank you for your interest. Can you send more details on your system and data thus far? The meeting. 2 days later, he found himself in a sunlit loft near University City. The exposed brick walls lined with whiteboards and strewn with circuit boards, tubing, and glass vials. Leah Montgomery, tall and soft-spoken with a cascade of auburn curls, greeted him first, shaking his hand firmly. Ethan Price, stocky, with a quick smile and paint-smeared jeans, gestured to a long desk piled with CAD prints. Hunter Caldwell, lean and intense, hovered near a whiteboard covered in flow rate equations. Dr. Greenham, Leah began, her voice warm, we know you've been working alone for too long. We've replicated your capsid expression in our microfluidic channels and increased yield by 40%, on top of reducing shear stress on the vectors. She tapped a tablet to display time-lapse footage of viral particles exiting narrow channels in perfect uniformity. Ethan chimed in, we built this array modular so you can scale up from a single 16-channel chip to a factory-scale 512-channel manifold without rewriting any code. His enthusiasm was contagious. Hunter pointed at a graph. And here's our purity analysis. 95.3% monodispersity, and it holds steady after 72 hours. Marcus felt a flicker of excitement, but also caution. This is impressive, he admitted, massaging his chin. But why come to me? You must have options at any large company. Leah exchanged a glance with Ethan and Hunter. We got tired of bureaucracy. At Gen X Biologics, every prototype had to pass 1,000 committees before anyone would touch it. We saw your paper and thought, here's work that actually matters, that could change lives. She paused. And honestly, because you're not the typical face we see in biotech leadership. We want to be part of changing that. Building trust. Those last words made Marcus's heart tighten. He recalled the countless meetings where raised eyebrows greeted him, the silent judgments when his boldest theories surfaced. He studied their faces, They were earnest, no condescension in their eyes, only respect. He spent the next hour quizzing them on every technical detail: pump calibration, channel dimensions, software interface for real-time monitoring. They answered with precision, occasionally admitting they'd hit snags: bubble formation in the T-junctions, minor clogging after extended runs, but showed clear plans to resolve each. When Marcus challenged their kill switch integration, Hunter sketched out a layered filter protocol that would remove residual promoter activity post-production. By the end of the meeting, the afternoon sun slanted through the loft windows, gilding the machinery in a hopeful glow. Marcus closed his notebook and leaned forward. "I won't lie, I'm wary. I've had offers before that evaporated when I needed support most." His voice softened. "But I've also been stubbornly working alone. Maybe it's time I learned to trust." Leah smiled. "We're not asking you to let your guard down. We're asking for a partnership built on mutual respect." She extended her hand. "If you'll have us." Marcus hesitated a heartbeat, then clasped her hand firmly. At that moment, the barriers between genius in the shadows and privileged engineers began to dissolve, united by the promise of discovery and the chance to outsmart prejudice together. The first uninvited allies had arrived, and with them, a new chapter in Marcus's fight for a viral cure that knew no color. By the end of April, an unassuming warehouse on the edge of Conshohocken had been transformed into a makeshift biotech incubator. Through its large garage-style door, Marcus and his three allies wheeled in benches, sterile hoods, and a gleaming bank of refrigerators. The space smelled of fresh paint, isopropanol, and promise. Sunlight streamed through high windows, illuminating rows of clear tubing, microfluidic chips, and Marcus's battered leather notebook, now filled with schematics and annotations in 3 colors of ink. Leah Montgomery took the lead on layout. She measured and remeasured the distances between each laminar flow hood and the sink, optimizing workflow to minimize cross-contamination. "Marcus, if we shift the reagent prep station over here," she said, tapping a drawing on her tablet, "we can reduce your walking time by 20 feet, and that's a minute saved every protocol." Marcus watched as she sketched out the adjusted footprints, nodding in approval. It was the first time he'd felt this level of professional respect. She valued his time, his effort, and his safety. Meanwhile, Ethan Price and Hunter Caldwell assembled the core of their innovation: a modular microfluidic bioreactor array encased in transparent plexiglass. Over the next few days, they calibrated each channel's flow rate using precision syringe pumps, patiently adjusting pressure regulators until each of the 32 parallel lanes delivered exactly 2.4 microliters per minute. At Marcus's insistence, they implemented an automated feedback loop, built with open-source code and off-the-shelf sensors that monitored particle size in real time. A fault-tolerant subroutine would pause the run and alert the team if any channel deviated outside the 0.1 micron threshold they'd set. Late one evening, Marcus loaded the first full production run: his novel capsid-engineered viral vector mixed with cargo plasmids. He watched, eyes bright behind safety goggles, as the fluid threaded through each channel, droplets forming with crystalline precision. On Leah's command, the sensors hummed, logged data, and plotted histograms, showing a monodispersity index of 94.7%, already surpassing their initial targets. Cheers broke the usual laboratory hush. For the first time, Marcus felt the weight of isolation lifting. He wasn't alone, but progress was never linear. A week later, an ominous stutter in the flow curves signaled trouble. One channel stalled, droplets coalescing into irregular beads. Ethan traced the issue to an air bubble lodged at a T-junction. "I must have over-tightened the fitting," he confessed, flushing. Marcus crouched beside him under the bench, shining a headlamp on the tubing. Together, they replaced the section, flushed the line, and reran the code to purge residual air. The mishap cost them an entire day, but it also solidified their trust. They celebrated by ordering late-night pizza and swapping stories of past lab disasters, laughter dancing above pipette tips. By mid-May, every facet of the process had been honed. Temperature controllers regulated channel heat, to within zero point 0.2 degrees Celsius. Custom-made magnets held reagents in place during automated wash cycles. Waste routing valves shuttled effluents safely into sealed biohazard bins. Marcus spent early mornings teaching undergraduates basic virology techniques, dictating step-by-step protocols, now printed and bound in a simple binder. His first official standard operating procedures. He watched as students handled viral preps with confidence, eyes alight with the same fervor he'd felt when he first peered through a microscope. Their crowning achievement came unexpectedly. During a routine run, Leah's real-time analytics flagged an unprecedented plateau in vector yield, 25% higher than even their optimistic projections. They clustered around the monitor, exchanging stunned grins. Marcus punched the air. "This is it," he whispered. "This is what we dreamed of." That evening, as the team locked up, they stood together under the warehouse's exterior lights. The night air carried the distant hum of highway traffic, a reminder of the world waiting outside. Marcus looked at Leah, Ethan, and Hunter, feeling a surge of gratitude. These once uninvited allies had become his family in science. In a space born of skepticism and prejudice, they'd built something far greater, a laboratory of hope where ideas mattered more than identity, and where collaboration could outsmart every barrier. With protocols validated and yields soaring, Marcus finally allowed himself to believe in the possibility of a cure, and of a world that might one day judge scientists by the content of their character and the strength of their data, rather than the color of their skin. The clock on the warehouse wall glowed 2:17 a.m. when Marcus Greenham finally allowed himself to slump back in his chair, exhausted but exhilarated. For weeks, the team had been racing against time and against doubt to refine their viral vector's stability and safety profile. Data had trickled in on uneven waves, promising spikes of efficiency, followed by baffling dips in yield. Tonight, however, felt different. All afternoon, Leah had recalibrated the automated microfluidic array's sensors, fine-tuning the gain on each photometric detector so that fluctuations of as little as 0.05 microns in particle diameter would trigger an alert. Ethan spent hours rewriting the control software to integrate a machine learning subroutine that predicted bubble formation before it happened, based on subtle pressure changes. Hunter, poring over the genetic sequence, had designed a novel microRNA scaffold that would further tighten the vector's kill switch performance. At midnight, Marcus loaded the next batch of produced vectors, each vial carefully labeled Run 0531, into the high-speed centrifuge for final purification. Under the dim red light of the cold room, he watched as samples spun down, the conical tubes whirling in a blur. His heart thumped. If they could clear the 90% purity threshold tonight, they would have the gamma sterilization data they needed for the GLP study proposal. By 1:02 a.m., the centrifuge's hum slowed to a halt. Marcus donned his gloves and goggles, then gently decanted the supernatant, isolating the viral pellet. He pipetted it into a sterile buffer and carried the tubes back to Leah's station, where she awaited with the NanoSite nanoparticle analyzer. "Ready?" she whispered, her voice echoing in the cavernous lab. Marcus nodded, hands trembling with anticipation. Leah loaded the sample into the flow cell. The machine clicked on, projecting live data onto the screen. Ethan hovered over her shoulder, laptop open to the custom script that parsed the raw size distribution curves into a real-time histogram. Seconds felt like hours. Then, the first bar lit up at 110 nanometers, squarely in their target range. A shower of data points followed, clustering tightly around that mark. The histogram's bell curve sharpened, centered at 112 nanometers with a polydispersity index of 0.07, well below their 0.1 cutoff. "Holy, that's better than we've ever seen," Ethan breathed, eyes wide. Hunter punched in a command to run the secondary purity assay, an HPLC run that quantified residual host cell proteins. As the chromatogram traced its peaks, Leah and Marcus exchanged a furtive glance. When the readout confirmed 92.4% viral particle integrity and under 0.02% contaminant proteins, Leah let out a whoop of triumph. They erupted around the bench. Marcus high-fived Hunter, Leah danced a quick jig, and Ethan pumped his fist. Their shared fatigue dissolved into pure jubilation. In that moment, the dim warehouse felt less like a makeshift incubator and more like the birthplace of a revolution. Yet they knew the real test lay ahead. If they could replicate these results consistently over multiple runs and demonstrate the safety kill switch in action, they would be ready to unveil their progress to potential backers. Marcus opened his laptop and began composing a brief, urgent email. Subject: Urgent Demo Invitation. Viral Vector Breakthrough. 2. Dr. Rachel Whitaker, Harrington and Cole. Samuel Jacobs, Broadstone Ventures. Dear Dr. Whitaker and Mr. Jacobs, we have achieved sustained 90% plus purity and exceptional monodispersity in our latest production runs, along with validated kill switch functionality in vitro. We would be honored to demo these results for you in person at our facility this week. Please let me know your earliest availability. Best regards, Doctor. Marcus Greenham. He hit send at 3:14 a.m. Almost immediately, his phone buzzed. A calendar invitation popped onto the screen. Demo at 2 p.m. Thursday. Harrington and Cole Capital Partners. They had accepted. The dawn light filtered through the warehouse windows as the team finally powered down equipment. Over slices of stale pizza and mugs of strong black coffee, they recapped every detail of the night's runs. Marcus studied their faces, bright with excitement, free of the skepticism that had shadowed earlier meetings. He felt a swell of gratitude. These white engineers had not only believed in his vision, but had brought it to life with their own ingenuity. As the sun rose, they gathered for a group photo beside the microfluidic array, its plexiglass panels still streaked with condensation from the overnight run. Marcus set the self-timer on his phone. Leah draped an arm around his shoulder. Ethan and Hunter leaned in close. The shutter clicked, capturing 4 scientists united by a common purpose. That afternoon's presentation would determine the fate of their project. But in the luminous calm of early morning, Marcus allowed himself to savor the victory. A genuine, hard-won breakthrough born not in the marble halls of Wall Street, but in the glow of a lab in the outskirts of Philadelphia. They had outsmarted every doubt. Now it was time to change the world. The polished mahogany table in Harrington and Cole's Midtown Manhattan boardroom reflected the late morning light as Dr. Marcus Greenham set up his laptop and samples. Across from him sat Rachel Whitaker and Samuel Jacobs, their expressions guarded. Andrew Thorne hovered behind them, arms folded, awaiting the demonstration. Marcus's pulse thrummed in his ears, but he spoke with calm authority. Thank you for coming. Today I'll show you not only our viral vector's performance, but also why diversity of thought and of team is crucial to innovation. He began by loading pre-prepared aliquots into the nanoflow cytometer. As the machine whirred, he narrated the process, how Leah's precise fluidics had elevated monodispersity, how Ethan's predictive software had preempted errant bubbles, and how Hunter's microRNA scaffold had tightened safety controls. The live data streamed on the projection screen, a bell curve centered at 112 nanometers with a polydispersity index of 0.06, and a kill switch activation assay showing 100% shutdown within 8 hours of inducer addition. Rachel's lips parted in surprise before she masked her reaction with a nod. Impressive, she conceded, but it's still lab scale. How can we be sure this translates to GMP production? Marcus clicked to the next slide, a time-lapse video from their Conshohocken facility showing the modular 128-channel array running uninterrupted for 48 hours. He explained how their standardized SOPs, now in version 3.2, ensured reproducibility across batches, and how they'd already pilot-tested a 1-liter preparation with consistent yields. As the video concluded, Rachel leaned forward, unconsciously smoothing her blazer. Samuel cleared his throat. Your team's technical merits are clear, but part of our mandate is assessing risk, especially reputational risk. The industry still expects certain pedigrees. At his words, The room's atmosphere shifted back to its earlier chill. Marcus met Samuel's gaze steadily. "I know what you mean," he replied. "But let me tell you about the people behind this data." He introduced Leah Montgomery, who had flown in that morning via video conference. Leah unspooled a brief recounting of her own struggle, how her designs were once shelved at Gen X Biologics for being too unconventional, until Marcus's vision gave them purpose. Next, Ethan appeared on screen, his workshop's backdrop cluttered with circuit boards and coding printouts. He spoke of being passed over for senior roles because he wasn't business enough, until, working with Marcus, he learned that true success lies in results, not titles. Finally, Hunter, camera trained on his cluttered desk, described the moment he first realized data and safety could overcome any bias. As each engineer spoke, the investor's skepticism visibly softened. The veneer of corporate detachment cracked, revealing genuine respect. When Leah signed off, the room was silent except for the faint hum of the projector. Marcus closed his laptop and said quietly, "Innovation doesn't come from echo chambers. It comes from bringing together people who challenge each other, and the system that holds them back." Andrew Thorne set down his pen and exhaled. "Dr. Greenham, this is more than a pitch. It's a blueprint for how biotech must evolve." Rachel nodded. "You've given us data, yes. But more importantly, you've shown us why we need to invest in underrepresented talent. We can't afford to miss out on the next breakthrough because of prejudice." Samuel clicked his fingers, and the senior partner rose. "We'd like to lead your Series A round," he announced, extending a hand to Marcus. The room erupted into polite applause. As Marcus shook hands with Rachel and Samuel, he felt the weight of every dismissed proposal, every joke at his expense, lifting. Later, back in the Conshohocken lab, Marcus gathered Leah, Ethan, and Hunter. He held up the signed term sheet. "Today you taught Wall Street a lesson," he said, voice thick with emotion. "Not just about science, but about respect. Let's never forget how hard we fought, for our data and for each other." They stood together under the harsh fluorescent lights, grinning and exhausted. In that moment, the line between ally and team dissolved completely. Together, they had transformed prejudice into progress and paved the way for cures that would benefit all of humanity. 3 months after that fateful boardroom meeting, the Conshohocken facility hummed with a new energy. The once modest 128-channel microfluidic array had grown into a row of 3 identical systems, each running round the clock under automated supervision. Stacks of bar-coded vials, packed with the capsid-engineered vectors, lined the stainless steel shelves, awaiting shipment for non-human primate studies. Marcus Greenham strode down the central aisle beside Leah, clipboard in hand, as red LED indicators blinked green in steady rhythm. "We've completed 12 consecutive runs above 90% purity," he reported. Leah smiled, tapping her tablet to display the latest QC metrics. And the in vivo safety data from Dr. Chen's team at the Primate Center came back clean. No off-target expression, full inducible shutdown on day 7. Meanwhile, Ethan and Hunter coordinated the next scale-up phase. In the nearby CGMP suite, they oversaw a pilot 10-liter batch under sterile conditions. Ethan adjusted the bioreactor's agitation speed based on real-time viscosity readings, while Hunter fine-tuned the reagent feed algorithm to maintain plasmid concentration. Every 6 hours, a courier collected samples for endotoxin testing and viral titration. By the end of May, with FDA pre-IND guidance letters in hand, the team filed their investigational new drug application. Marcus stayed late that Friday, drafting the cover letter. "Herein we present comprehensive preclinical safety and efficacy data, enabled by a multidisciplinary team that defies convention." In mid-June, Marcus's inbox blinked with an invitation that felt surreal. TEDx Boston would be honored to feature your talk on innovation and inclusion in biotech. He read the email twice before forwarding it to Leah, Ethan, and Hunter. "We made it," he wrote. On July 15th, he stood backstage at the Wang Theater, nerves coiling in his stomach. The house lights dimmed and the red circle of the TEDx logo glowed on the wooden floor. As he stepped into the spotlight, he felt the warm hush of 1,200 expectant faces. He began with a story of a young boy in West Philadelphia, poring over science textbooks by the dim glow of a desk lamp, told he was different before he even spoke his first word of science. He introduced his allies, Leah, the fluidics savant, Ethan, the code whisperer, and Hunter, the genetic architect, calling them up on stage one by one. Each engineer spoke into the handheld mic, sharing a sentence about how collaborating beyond color and hierarchy had led to a cure nobody believed possible. The auditorium erupted into applause, so thunderous, Marcus had to pause, blink back tears, and simply breathe. After the talk, reporters and fellow scientists swarmed the stage. A journalist from Nature Biotechnology asked, "What's next for Greenham Biotech?" Marcus answered steadily, "Phase 1 human trials, funded by our Series B round, beginning this fall. But beyond that, I want to see a world where talent from every background can thrive, where no brilliant mind is sidelined by prejudice." The phrase "prejudice is the greatest pathogen" quickly became a hashtag trending on Twitter. Back in Conshohocken the following week, the team gathered around a conference table to open a congratulatory letter from the National Institutes of Health, a $15 million grant to support global trials in underserved communities. Leah tore open the envelope, her eyes shining. Ethan punched the air. Hunter looked on, arms crossed, a rare grin spreading across his face. Marcus held the letter aloft and said, "We did it. We showed the world what's possible when you let data and people lead." That evening, after the celebration dinners and media interviews, Marcus returned to his childhood home in West Philadelphia. On the front porch sat his mother in her favorite rocking chair, knitting a bright yellow afghan. She looked up with a knowing smile. Marcus knelt beside her, gently kissing her cheek. "We changed things, Mom," he said softly. She touched his hand. "I always knew you would." As dusk deepened into night, Marcus gazed down the tree-lined street where children played. He thought of the years spent in labs, of every dismissive glance, every locked door. And now, as the Afghan unraveled in his mother's hands, he felt the story close its final loop. A viral cure born from brilliance and brotherhood, opening doors for the next generation, no longer talent in the shadows, but champions in the light.